Can everyone see my slide okay? Awesome. Yeah, thanks for that really nice introduction. And also thank you for the organizers for putting together this webinar series. And it was also really nice to see a few familiar faces at the beginning of the seminar. So it's really nice to see everyone. And thank you all for tuning in today. I know that we're all calling in from different places and it's early for some people and late for others, but I really appreciate people tuning in today. Today, I'm going to be talking about how enhanced substrate stress relaxation promotes filopodia-mediated cancer cell migration. My name is Kolade Adebowale. I'm currently a postdoctoral fellow in the Mitragotri lab at Harvard. But the work that I'm actually going to talk about today was for my PhD studies working with Professor Ovijay Chowdhury at Stanford University. Cell migration is an essential biological process that's important for embryogenesis, wound healing, immune cell trafficking, and cancer metastasis. What happens during metastasis is that cancer cells break away from a primary tumor, they migrate through an extracellular matrix or ECM, they enter a blood vessel, exit that blood vessel, and then colonize a secondary site. This process of metastasis is responsible for up to 90% of cancer deaths. And so many groups around the world have developed biomaterials to mimic certain properties of the ECM and to try to understand how those properties impact cell migration. For example, some groups have looked at the role of mechanical confinement on cell migration. Others have looked at how adhesion impacts cell migration, or how differences in Young's modulus impact cell migration. However, the potential impact of viscoelasticity on cell migration is not known. It turns out that normal and diseased tissues are viscoelastic and they exhibit substantial viscosity. And this is demonstrated by this graph on the left, which is looking at the loss modulus, which is a measure of viscosity on the y-axis as a function of the storage modulus or a measure of elasticity on the x-axis. And there's a lot of data on this slide, but I'd just like to point out that there's a diagonal line and a lot of the data are clustered around this diagonal line. And what that means is that the data that are along this diagonal line are viscoelastic materials. So they have both properties of fluids and solids. And so normal biological tissues like brain, liver, and lung, these are all viscoelastic materials. In addition, diseased tissues like breast cancer are also viscoelastic. And this is shown by this graph on the left, which is looking at the loss modulus on the y-axis as a function of the dynamic modulus on the x-axis. And what the data shows is that the malignant human breast tumor samples are not only stiffer, so that's the data shown in red, but they also have a higher loss modulus or viscosity compared to benign tumors. And this increase in both stiffness and loss modulus has also been found for bladder cancer, liver cancer, and pancreatic cancer. One of the consequences of viscoelasticity is that these materials exhibit what's called stress relaxation. And we can investigate this in the lab by doing a stress relaxation test where we input a constant strain into the material and then measure the stress response. And here's a schematic view of what the result from that experiment looks like, where we're looking at the normalized stress in the material as a function of time. For a purely elastic material, like a rubber band, there's no change in the stress in the material as a function of time. In contrast, however, for a viscoelastic material, for example, like Jell-O, the stress in the material decreases as a function of time. Just to quickly remind you, Lamellipodia are thought to be a universal mediator of 2D cell migration. Lamellipodia are these structures that are found at the front of a cell that's migrating. They are characterized by a dendritic actin network, cable-like actin stress fibers, and a very spread cell morphology. And we currently have a textbook view of the critical role that Lamellipodia play during cell migration. And that's highlighted by this paragraph on the bottom that says that cells that are not able to make normal Lamellipodia crawl poorly and tend not to metastasize. Over the last several decades, there's been intense focus in the biomaterials community to understand the role of stiffness on cell migration. What's been found is that if cells are on a compliant substrates, shown over here, the cells are very rounded and they're non-motile, which is shown in this white region. But if the same cells are on a more rigid substrates, those circular cells, they have a very spread morphology and they acquire a motile phenotype shown by this light blue region. And that associated with this transition is the formation of these Lamellipodia structures at the front of the migrating cell. Of note, however, most of these studies use very rigid or purely elastic substrates. In this project, however, we develop hydrogels with independently tunable stress relaxation properties. This hydrogel consists of two main components. The first is basement membrane matrix, which a cell can bind to and recognize. The second component is alginates, which is shown by these dark blue lines, and it's ionically cross-linked by adding calcium ions. The way that we modulate the stress relaxation behavior is by changing the molecular weight of the alginates. That then allows us to make a range of viscoelastic materials that all have the same initial Young's modulus of about 1.5 kilopascal, or a purely elastic polyacrylamide hydrogel that has the same initial Young's modulus. These materials are quite compliant. So to give you a sense of how compliant they are, a gummy bear, for example, has a Young's modulus of 70 kilopascal, while these materials have an initial Young's modulus of 1.5 kilopascal. In any case, however, we can quantify differences in stress relaxation behavior for the viscoelastic materials by doing this stress relaxation test that I introduced earlier. And we can, for example, say that the time that it takes for the peak stress in the material to reduce to one half its maximum value is a characteristic time called T1/2. And that number is about 100 seconds for what we call a fast stress relaxant hydrogel, or about 2000 seconds for what we call a slow stress relaxant hydrogel. To establish some nomenclature for the rest of this talk, whenever you see fast or slow, that refers to fast or slow stress relaxant hydrogel. We like this material system because it allows us to tune the stress relaxation properties while maintaining similar Young's modulus, constant matrix composition, and similar pore size. This is important because later on, if we observe any differences in cell migration, we can attribute those differences to differences in stress relaxation. So now we would like to know, what is the impact of differences in substrate stress relaxation on 2D cell migration? To study this, we put cancer cells, for example, HT1080 cells, on top of these hydrogels and watch how they migrate. For example, here we see a cancer cell that's migrating from left to right. We can then visualize these differences when the cells are migrating on either a fast relaxing or slow relaxing substrates. So each of these lines you're looking at is the trajectory of an individually migrating cell, and they're all plotted on a common axis for easy visualization. And here's a scale bar to give you a sense of how far the cells are migrating. And what I hope you can at least appreciate qualitatively is that the cells are more migratory on a fast relaxing compared to a slow relaxing substrates. Furthermore, we can quantify these differences, for example, by looking at the cell migration speed. And what we find is that the cells have a higher cell migration speed when they're on a fast relaxing compared to a slow relaxing substrates. We've also obtained similar results for MDA, MB231 cells and MCF10A cells. So these results are not cell line specific. We also find that fast substrate stress relaxation potentiates a super-diffusive migration behavior. And we quantify this by looking at the mean square displacement, or MSD. So the MSD is a metric used to assess how efficiently a cell is migrating. The higher the MSD, the more efficiently the cell is migrating. And what the data shows is that the cells on the fast relaxing substrates have a much higher mean square displacement compared to the cells on the slow relaxing substrates. It turns out that this MSD can also give us a sense of the underlying migration behavior by extracting what's called an alpha coefficient value. And if this alpha value is greater than 1, as in the case of the fast relaxing hydrogel, we have a super-diffusive migration behavior. If the alpha value is less than 1, as in the case of the slow relaxing substrates, we have a subdiffusive migration behavior. And so in the interest of time, because we observe the most robust cell migration when the cells are on fast relaxing substrates, I'm going to only focus on the results from those experiments going forward. One of the things we observed was that when we looked at the edge of these cells on, say, a fast relaxing substrates, we observed these finger-like extensions protruding from the cell membrane, and these are highlighted by these yellow arrows. These protrusions contain a protein called actin protein. And we observe these protrusions when the cells are on a fast relaxing as well as on a slow relaxing substrates. In contrast, however, we observe a different kind of actin structure called Lamellipodia when the cells are on a more rigid substrates. And so together, this data suggests that Lamellipodia are not observed on stress-relaxing substrates, but we observe a different kind of actin structure. We also wanted to know if these protrusions have anything at all to do with cell migration, and it appears as though they do. So here we see a cell that's migrating from left to right. As the cell is migrating, we see protrusions form at the front of the cell, and these protrusions are highlighted by these images over here. It turns out that the number of these protrusions is very sensitive to the mechanics of the material, such that there are more of these protrusions when the cell is on a fast relaxing substrate compared to a slow relaxing substrate. These protrusions reminded us of what are called filopodia protrusions. So we stained these protrusions for a protein called myosin 10, which is a key marker of filopodia, and these protrusions indeed contain myosin 10 protein. Not only do they contain this myosin 10 protein, they also contain additional key filopodia proteins, which are called VASP and P130Cas. And so together, to summarize, the data demonstrates that the protrusions we observe are filopodia protrusions. To further confirm the role of filopodia in cell migration, we use the combination of small molecule inhibition and genetic knockdown to perturb the activity of key proteins involved in filopodia formation, and then ask the question, what is the potential impact of those perturbation experiments on cell migration speed? And so, for example, if we use small molecules to inhibit the activity of forming and fascin, these are two key proteins involved in filopodia formation. The ability of the cell to migrate is significantly diminished. If we use a more targeted approach, for example, by using siRNA to knock down either fascin 1 or myosin 10, these are again two key proteins involved in filopodia formation. The ability of the cells to migrate is significantly diminished. So together, this data suggests that filopodia protrusions and associated proteins regulate cell migration on fast relaxing substrates. It turns out that when these cells are interacting with substrates, they're able to form adhesions. And we use phosphorylated paxillin as a proxy for these adhesion structures between the cell and the substrates. And in particular, we're focusing on these circular small dot-like structures that are found at the periphery of the cell membrane called nascent adhesions. We observe these nascent adhesions when the cells are on a fast relaxing and slow relaxing substrates. Interestingly, however, these adhesions are also very sensitive to the mechanics of the material that they're interacting with, such that there are more of these adhesions when the cell is on a fast relaxing compared to a slow relaxing substrate. The size of these adhesions, however, is very similar regardless of the substrates. In contrast, if the cells are on a more rigid substrate, not only do we observe these dot-like nascent adhesions, we also begin to see evidence of what are called focal adhesions, which have a more elongated geometry. And these adhesions are also mechanosensitive, such that there is a dramatically higher number of adhesions in general when the cells are on a more conventional rigid substrate compared to the stress-relaxing viscoelastic substrates. So to summarize, for stress-relaxing substrates, fast stress relaxation promotes the formation of nascent or weak adhesions. when the cells interact with the substrates and adhere to that substrates, they're able to use those adhesions to generate traction forces to support cell migration. We can investigate this process by embedding fiducial markers, or beads shown in red in this image, inside the hydrogel and looking at how these beads are being displaced in response to cell migration. So if you look at the yellow squares, for example, as I play this movie, hopefully you'll see that the beads are being displaced in response to cell motion. We can then visualize this process, for example, by generating these kinds of heat maps, where the heat maps are color-coded such that the cooler colors indicate a low substrate displacement, and the warmer colors indicate a high substrate displacement. And the arrow gives you a sense of the direction in which a substrate is being displaced over time. We can then quantify the maximum amount of traction stresses that the cells are generating on either a fast relaxing or slow relaxing substrates. And what we find is that the cells have greater traction stresses when they're on a fast relaxing compared to a slow relaxing substrates. Furthermore, we find that the ability of a cell to migrate is significantly impaired if we inhibit the canonical traction force generation machinery within the cell. So for example, if we partially inhibit actin polymerization, or if we block myosin activity, or if we partially inhibit beta-1 integrins, doing either of those perturbations significantly decreases the ability of a cell to migrate. To obtain additional molecular insight into what's going on, we wanted to implement a motor clutch model of cell migration. And this is work that we did with our collaborators at Penn and also at Minnesota. This model consists of three main components. The first is actin polymerization occurs at the front of the cell, and this creates an actin protrusion. The second component is that there are myosin proteins that bind to that actin protrusion and pull that protrusion toward the cell body. And the third component is that there are clutch molecules, such as integrins, that the cell can use to interact with the viscoelastic substrates. The first thing we wanted our collaborators to do was to be able to reproduce some of the basic experimental observations that we found. And we were really excited when their simulation showed that the mean square displacement is much greater when the cell is on a fast relaxing compared to a slow relaxing substrate. So these trends match very nicely what we had observed experimentally, and it gave us a lot of confidence in this model. We then wanted our collaborators to make some additional predictions that we can test experimentally. So in this movie that I'll play in a second, you'll see these filopodia protrusions highlighted by these red arrows. They have a kind of dynamic behavior, so they can extend and they can retract over time. And the simulations predicted that these filopodia protrusions, their length and their lifetime would both be greater when the cell is on a fast relaxing compared to a slow relaxing substrates. And we were really excited when we went into the lab to test the simulation predictions. And what we found experimentally matched very nicely what was predicted by the simulations, which is that both the length of these protrusions and the lifetime is very sensitive to the mechanics of the materials they're interacting with, and is actually much greater when the cell is interacting with a fast relaxing compared to a slow relaxing substrates. So to briefly summarize, we propose a model which is shown by this schematic over here, where a cell is sitting on top of a viscoelastic substrates. And if we focus in on this interface, for example, between the cell and the substrates, there are three main components. The first is actin filament shown by this green rod. The second component is the substrate shown by this brown rectangle. And there's a mechanical connection, which are these springs, such as integrins, that connect the inside of the cell, the actin rod, to the outside world, the brown substrate rectangle. And because these substrates are stress relaxing, their modulus actually changes over time when they're being pulled on, for example. And this is color coded such that the initial Young's modulus is a darker brown color, and the long-term Young's modulus is a lighter brown color. But at the same time, this is the mechanical connection, and this rod is being pulled to the left. And as it's being pulled to the left, the spring comes under load. And this spring, such as which represents integrins, for example, is color coded such that the cooler colors indicate a low bond force and the warmer colors indicate a high bond force. So these springs or integrins get stretched, they come under load, they come under more load. But again, because this is a stochastic process, this mechanical connection at some point is disrupted. And this process repeats itself over and over again. And this is what happens when the cell is interacting with a fast stress-relaxing substrates. In contrast, however, when the cell is interacting with a slow relaxing substrates, the mechanical connection is abruptly disrupted, and this prevents the cell from being able to generate sufficient traction forces to support cell migration. So perhaps one way to think about this is to think of the case of a fast stress relaxant hydrogel as pulling on a slinky, and the case of the slow relaxant hydrogel as pulling on a car strut. And if you're pulling on a car strut, the bonds are loading up very quickly, but they're also failing very quickly. In the case of a slinky, however, it takes time to build load on a slinky. And because you have that time, that allows you to recruit all the proteins necessary to form filopodia protrusions and then generate traction forces to support cell migration. So to summarize, what I hope I demonstrated was that we develop materials with independently tunable stress relaxation properties. We can quantify the differences in stress relaxation by doing a stress relaxation test. We can then put cancer cells on top of these substrates and watch how they move around. And we currently have a canonical view that cells are non-motile when they're on a compliant substrates. In contrast, however, in the current work, we demonstrated that even if the cells are non-motile on a compliant substrate, if that same substrate has sufficient stress relaxation properties, those non-motile cells can transition to a very motile phenotype shown by this light blue region, and that associated with that transition is a formation of filopodia protrusions at the front of the cell, and this drives cell migration. And with that, I'd like to thank Professor Ovijay Chowdhury, my PhD mentor, all the members of the Chowdhury family, our numerous collaborators, our funding sources, and I'd love to connect with you all on Twitter. And I know that some people may be watching this video later on, so please, if you have any questions, please feel free to email me at kaa@g.harvard.edu. Thank you for listening, and I'll be happy to take any questions now.
Thank you very much for Kolade's excellent presentation. So any questions from the audience, and please introduce yourself before your question. You can click the, in the Zoom option, we have the raise my hand, or you can just unmute yourself, ask directly. So any questions? If no one asked, I have
a question, sorry.
Okay. Oh, sorry. Please go ahead, Jing.
Hi, colleague. Very nice talk, very interesting. We know that the biological system much more complex than a simple model of adhesion or cohesion with interface. I was wondering, like, what is the speed of the cancer cells moving on a rigid subject compared to the hydrogels? I don't recall there's a speed comparison in your slide.
Yeah, that's a really good question. So let me go back here. So yeah, so let's look at the graph on the far right, for example. So for the glass substrates, it is, I'll say it's somewhere between the blue bar and the orange bar. So it's actually a little bit less, interestingly, than when the cell is on a fast relaxing substrates. Yeah.
I see. That's very interesting. So that's why I was wondering, like, because we know glass, if it's just regular glass, doesn't been treated with hydroxyl groups or different groups, they won't have a strong hydration layer compared to the hydrogels. So I was wondering the intrinsic adhesion between the cells, as you show that they have some differences on the cell size, but I was wondering on the material size, is there any difference between the hydrogels and the glass will play a role in the speed or in the motion of the cells.
Yeah, I think it definitely does. So let me see if I can escape this. So I think definitely the intrinsic differences in the materials definitely plays a role in the differences in cell migration. So also one thing to quickly point out is that if we look at this data, let's say we look at the this data over here on the top, and then compare it to this one on the bottom. The thing is that when the cell is on a glass substrate, for example, it has more adhesions. So because it has more adhesions, it's actually a little bit slower than if it has fewer adhesions. It's a little bit counterintuitive. It's almost like a fly on a fly trap. Because the fly trap is so sticky, it has so many adhesions, it actually can't move very well. So there's a sweet spot where you want some adhesions, but you don't want too many adhesions. And some adhesions, this intermediate sweet spot is what actually really drives promotes the cell migration. So yeah, it was really surprising to us when we found that the cells are actually, they have higher migration speeds on these fast relaxing substrates.
Yeah, exactly. The more adhesion, maybe like it's more sticky, but it moves faster. That's fascinating. Yeah, thank you.
Yeah, no problem.
Yeah, Kolade, my question is, I saw many other people like Jacob Noteboom at Wisconsin and Daniel Cohen at Princeton. They do those like a collective cell migration problem. So do you have some comment like for cell problem? Do we need to look at a single cell? Or do we need those like a multiple cell migrations?
That's a great question. I think it depends on what context you're looking at. I think for things like embryogenesis, for example, I think probably collective cell migration is a little more relevant. I think for cancer, for example, which is what this talk is about, I think my sense is both are important. So some cancers disseminate when they metastasize as clusters. And so for those specific cancers, it's very important to focus in on collective cell migration. But there are also other cancers that when they metastasize as individual cells. And so for those cancers, it's also important to think about single cell migration. So I think in terms of cancer, it depends, both are important, both collective and single cell migration. Yeah.
Thank you, Kolade.
Yeah. And I think it's only now that we're getting better at imaging, for example, with like intravital imaging and in vivo imaging, that we can really begin to visualize which specific cancers are disseminating as clusters and which are disseminating as individual cancer cells. And I really anticipate that over time, we're going to have a much better sense of what's the relative contributions of collective versus individual. Does it depend on the stage of the cancer, even for the same cancer, or what are the different factors that determine what mode of migration we'll observe? Yeah.
Thank you, Kolade.
Please go ahead, Altman.
Great. Hi, Kolade. Yeah, really great talk. So I don't know, maybe a naive question, but I was just sort of more curious about the differences between filopodia and Lamellipodia, right? So the mechanisms that you described at the end of your talk, would you also expect that same thing to be happening in Lamellipodia? For example, basically that mechanism. And then just another really basic question, is the structure of the two morphologies the same? So for example, the actin within those structures, is that same level like actin expression, that kind of thing?
That's a great question. So let me take the second question first. So basically, for the Lamellipodia structure, so in this cartoon on the bottom right, these green things, these are actually the actin protein. So the actin structure is very different between filopodia and Lamellipodia. So with the filopodia, they're almost like it's almost like a finger from my hand. They're kind of like spiky, and they're actually protruding visibly from the cell membrane. In contrast with the Lamellipodia, it's more of a mesh, it has a very stereotyped architecture that's shown by this dendritic structure on the bottom right over here. So this actin structure is very different between those two. And the proteins that mediate these different actin structures are also very different. So for filopodia, for example, things like farming are very important. Whereas for things like Lamellipodia, ARC23, which is another completely different protein, is very key for the formation of Lamellipodia protrusions. So the structures are very different. And then the overall, just the gross cell morphology is also very different. So on the stress-relaxant substrate, the cells are very, very rounded. Whereas on the rigid substrates, when they form Lamellipodia, it's more like a pancake. They're flat out and they spread out a lot. And then to your first question about the mechanism of what's happening. So
sorry, before you go, so by rounded, you mean in 3D as well, right?
Yeah.
Not spherical. Okay, cool.
Yeah, so basically, if you look at this schematic on the top left, they kind of ball up a little bit. Whereas if you were on so this is representing a viscoelastic substrates. If this were a glass, for example, they'll just be flatter on that substrates. Yep. And then to your first question about the mechanism as to what's happening on, I think you're referring to this slide.
Yeah. So basically, So basically, would you be observing the same thing in a Lamellipodia morphology, cell morphology?
That's a great question. So we haven't tested that, but I assume we may see something very similar if the cell is forming Lamellipodia on substrate with different stiffnesses as well. Mm-hmm. So I think we may be seeing something similar as well. Yeah, that's my best guess.
Thank you.
Yep.
Thank you, everyone. So due to the time limit, let us move to the second speaker. Kolade, could you please share your screen? So
thanks.
And our second speaker is Haozhou. He will talk about the accelerated computational micromechanics and its application to polydomain liquid crystal elastomers. Hao, could you share your screen? Dr. Haozhou obtained his PhD degree from Department of Mechanical Engineering at Caltech, working with Professor Akashik Bhattacharya. He received his bachelor's degree from Peking University in 2016. His research focuses on the development of computational micromechanics algorithms and the behavior of soft materials. And now the stage is yours.